another edition of the Lab 207 webcast. My name is Mr. Kite, and I will be hanging out with you today as we continue on in our very long series about the origins of life. The topic for the day is changes in body form. So how did we actually get different body forms throughout evolutionary history? Here's your objectives for the day. So by the end of this video, you should be able to understand mechanisms by which major changes in body form can arrive or arise. That probably should have been arise. Second thing, describe some of the mechanisms that may be responsible for novel body forms. So that's what it's all about today. And here's why we're talking about this. So we've talked about a lot during this series that there's ample evidence for the fact that evolution has actually happened, whether it's microevolution or macroevolution. We've shown pretty strongly, and science shows pretty strongly, that it does happen. But the big question is, how does it happen? We see in the fossil records that there are all of these jumps from one form of animal to the other, but where do these novel body forms and body plans and shapes and sizes, where do they come from? So that's what we're going to talk a little bit about today. So I'm just going to go ahead and jump on into some of the mechanisms by which scientists think that novel body forms can arise. First one is with the developmental genes, the rate and the timing with which they are expressed. I'm going to give you two examples. So the first one is called heterochrony. What that means is that different genes may be expressed at different times and at different rates, giving you a drastically different body form. I'm going to draw you just a generic little picture here. So let's say, for example, that this box represents the skull of a chimpanzee as a baby. Here's a human skull as a baby. I know that obviously we are not square, but I'm just using this to exhibit a point. So as this baby chimp grows up, different genes are going to be expressed affecting the shape of the skull. So for a chimpanzee, it might look like the back of the head gets expressed a little bit more, making our box go this way, and then maybe the jaw genes get expressed a little bit more, giving us a shape that's something like this. So as these genes were being expressed, and the extent to which they were being expressed caused an outgrowth in the back of the skull, and it caused the front of the face and the jaw to jut forward. Now, take those same genes in humans that control the rate at which the back of the head and the jaw develops and turn them on at a different time. Remember, it's all about words. Hetero means different, chrony means time. So turn these genes on and off at different times, and our human skull develops fairly symmetrically. All right, so instead of turning the genes on and overexpressing the genes that take care of the jaw and the back of the head like we do in the chimp, everything is expressed pretty symmetrically. So that is a way that even though they're the same genes, you express them at different times, you can get a totally different shape between the skull of a chimp and the skull of a human. The second one right here, paedomorphosis. Paedo is childlike, morphosis is form. What this is talking about is usually once an animal develops into its sexually mature state, it stops developing new features. It just grows and gets bigger. So there are some organisms that it is possible that they actually reach sexual maturity before they reach their mature form. So it's kind of like they looked like the kid form of their organism for a while, and then as soon as they became sexually mature, they stopped developing new features that would have taken them to the adult form. So these animals were essentially locked into a childlike form. They grew, they got bigger, but they still had the features of the juvenile organism of their species. So those are two ways that developmental genes, like the rate at which it's expressed, heterochrony, and the timing, the paedomorphosis, could have given us novel body forms. Next thing to talk about is spatial pattern. We've talked about homeotic genes. Those are the genes that control what goes where. We have talked about Hox genes, which, like on the right there, the same genes that controls where the torso goes in a human controls where the thorax goes in a fly. You know, it's about the same thing. If you take the genes and rearrange them, you can get novel body forms. There have been a lot of experiments done, and you see funny pictures in textbooks of Drosophila that have got legs sprouting out of their forehead because the scientists went in and mixed up their Hox genes to control what goes where. So simply by shuffling the deck of cards on those Hox genes, you can get totally different and novel body forms. Next up on the parade is gene changes. Now, one of the things scientists have looked at is how did we get from aquatic crustaceans to insects? And they did a lot of work. Poor little Drosophila, they're always getting work done. There's a gene called the UBX Hox gene that essentially controls how many legs either the crustacean form of an animal has or the insect form. And scientists did so much work on this gene that they were able to sequence it out, Then they artificially constructed this chromosome, and then they were inserting their artificially constructed chromosome into Drosophila. What they were trying to work out is what type of mutations would it take to go from this crustacean, which has like 20 legs, to an insect which has a much more practical 6 legs. They did so much work on this gene that they were able to isolate the single amino acid change needed in order to go from multi-leg form of this gene to a 6-leg form of this gene. So sometimes it just took a small single base or single amino acid change in a gene in order to move us from one form of an organism to the next. And this is the last one. Quick video today. We've talked a lot about gene regulation too, and the reason gene regulation is interesting is because you get change but you don't have the cost of mutation. So a lot of times mutations are detrimental to where they actually can harm the organism. If we're just talking about gene regulation, genes are there, they're not getting rearranged, it's just the way that they are being expressed is being affected. So these genes could be methylated, turning them off. They could get extra promoters or enhancers to turn them on more frequently, or they could just be expressed more fully. Example of this is there are many forms of these stickleback fish. Some forms, as you can see up there in A, on their pelvis, they have got very pronounced spines that jut out of their bodies, and obviously this is probably a defense tool. There's another form in areas that have fewer predators that the spines are pretty much absent or not apparent at all. So the genes, while still there, are being much more expressed in the form that needs the spines for defense than they are in the form that does not. So the genes are still there, but one form is being more expressed, the other form is being less expressed. So that's it. Quick whirlwind tour of possible ways that we could get novel body forms arising on the scene evolutionarily. Hope this was helpful to you. Thanks for joining us on the Lab 207 webcast. My name is Mr. Kite, and we'll see you again.